for 2026. And we've got lectures now running consecutive Tuesdays going forwards at 7 o'clock, so that's easy to remember. And as is our way with Liverpool Head and Neck Centre, we've got a real variety of topics and disciplines dotted in and around anything to do with head and neck. So we've got a psychology, an issue with quite a clinical talk about older adults and head and neck cancer. We've got a clinical trials talk about the interaction of robotic surgery and radiotherapy. So just to remind people, we do not continue every Tuesday through the year. We do little short terms of 4, 5, 6 lectures, and that's the way we do it. So you can just bear with us in January, February. You can have your summer months back, and we won't be bothering you in the early evening, so you can go out and enjoy the sunshine. It seems like impossible, doesn't it, that we'd have sunshine after we're properly into winter, aren't we? Stop sharing. So tonight, it's my enormous pleasure to invite Amy Harding to give her lecture. Amy's appointment was a huge success for the Head and Neck Centre in many ways. It came off a programme grant that we wrote collectively, and particularly with Professor Hunter, to try and drive the translational side of our research group forwards. So Amy's one of our newest recruits. She's a PhD scientist. She's a doctor, but a proper doctor, not one of our medical doctors. And we managed to steal her over from Sheffield. Her background was BSc in Sheffield, and her MSc and PhD were all in Sheffield. PhD was in roughly this topic that she's continued with, which was awarded in 2020. And since then, she's had postdoctoral positions in the oral cancer related research group in Sheffield. And over the last year or so, she's come over for a research fellowship to Liverpool Head and Neck Centre and continuing the work. And so I just wanted to mention that Amy's appointment has been made possible not only through the Head and Neck Centre, but also in particular through Northwest Cancer, through a programme grant about our capacity to drive translational research. So a good shout out to Northwest Cancer Charity. So with that, I'm going to hand over to Amy, and we can enjoy Amy's talk, which is titled Modelling the Extracellular Matrix in Head and Neck Cancers from Scaffold to Saboteur. Thank you, Amy.
Thank you so much, Richard. I'll just share my screen. I'm hoping that's going to be... Yeah, bear with me one moment. Is that visible? Right there, let's put it into...
So it's visible, Amy. If you just press play on the presentation, that's it. Perfect.
Yeah, all good. Okay. Right, guys, thank you so much. And thank you, Richard, for that kind introduction. I'm hoping to get you thinking a little bit about not the cancer cells tonight, but a bit of the meshwork that surrounds it called the extracellular matrix, and how it acts as a very active contributor to disease progression in head and neck cancers, as well as other solid cancers. Talking about how it has a major impact on shaping tumor invasiveness, immune behaviors, and as a translational scientist, clinically in the form of recurrence and therapeutic outcomes. I'll likely talk about my research framework and how I describe extracellular matrix states and ECM niches, but the lecture will largely cover today why the extracellular matrix matters in head and neck cancers and why I think you guys should take it into consideration. I'll talk about how we model it in the lab, looking at some of my research from my PhD, and lots of research here at the Liverpool Head and Neck Centre under the pathobiology group. I'll also talk about some of what these models have revealed to us so far and what we're hoping to use them for in the future. And like I said, I am a translational scientist, so all of what I do, I'm hoping to have some sort of translational outcome for patient benefit, and whether that's the form of biomarker discovery or combinational strategies. But the whole aim is to look at this data and how we can better help patients in the future. Okay, so you've heard this loads and you're probably sick and tired of hearing it, but I'll just reiterate the fact that depending on what stats you look at, head and neck cancers are probably the 6th, 7th most common cancer worldwide. Now, in the UK and in the Western world, we're seeing a projected UK incidence rise in particular subsets of head and neck cancers. However, overall, we're still seeing a pretty poor survival rate at 50% at 5 years. So there's still lots to be done. Even if our surgical intervention's improving and our therapies are improving, we're still seeing this really poor survival long term. And I think one of the major contributors to that is the fact that head and neck cancers arise in very, very distinct anatomical sites and locations. So these are all biologically distinct sites and subsites. So you can see that you get cancers of head and neck, including the oral cavity, so cancers of the mouth. You also get cancers of the oropharynx and different subsites of the oropharynx, as well as the larynx, and then other smaller subsites like salivary glands and nasosinuses. A core argument that I'll reiterate today is that we can't really understand the differences between these subsites and the impact that has unless we start to really look at subsite differences in the anatomy and the stromal compartments of each of those anatomies. What we find is that in between these different types of subsites, you get a very localized tissue context. So you get varying extracellular matrix compositions, architectures, mechanics. You get very, very different baseline properties and immune microenvironment differences in each of these subsets. So that really grounds the fact that we need to start thinking about this not as one disease, but as a collective disease, disease with lots of different subsite and microenvironment cues that have an impact. I also want to point out that this is quite a regional concern, and a big part of why the Northwest Cancer Research is funding this work, along with the Liverpool Head and Neck Centre, is we're seeing a disproportionate amount of oral cavity, oropharyngeal and hypopharyngeal cancers in the Cheshire and Merseyside region in comparison to that of England. So it's very interesting that we're seeing an increase in some of these subsites, and that tells us that we always have to look at these in very different contexts when we're talking about matrix and how that might influence disease outcomes. This just puts it into a little bit of perspective. It's a very, very sort of generalized summary, but I've just taken three sites in particular: the oral cavity, the oropharynx, and the larynx. And it's pretty easy to see that you get subsite heterogeneity where there's different drivers clinically of what's happening. So for example, in the oral cavity, we tend to see these sort of high stromal based tumors. So you get really myofibroblastic CAFs. These cancer fibroblasts are producing a lot of matrix components, and that's having a distinct impact on the immune environment within the oral cavity. So you tend to see lots of desmoplastic tumors within the oral cavity, which is different to other subsites, such as the oropharynx. You may not see this more desmoplastic ECM, but you often see disease associated with an HPV-driven disease. So you get lots of different lymphoid contexts, and that can then sort of impact how the stroma is into play in the oropharynx as well. And for the third subsite, just for example, is the larynx. Again, this has quite a distinct epithelium, so a very different anatomy to some of the sites, depending on what part of the larynx you're looking at. So you get a mixture of squamous cell carcinomas, but you also get the more columnar, pseudocolumnar epithelium as well, and that respiratory epithelium. And that can be largely linked to exposure history, such as smoking and alcohol. And we don't tend to see the HPV positive better outcomes like we do with oropharynx, like we do with the larynx. It doesn't really follow the same pattern. So that's something we need to really think about too. Okay, so what does this mean as far as extracellular matrix phenotypes go, and what do we know about them in head and neck cancers? So I just want to tell you what the ECM does in normal physiology if you know nothing about it. We have extracellular matrix throughout all our tissues, or all our organs will have some sort of extracellular matrix component. For the most part, it acts largely as a supportive scaffold. So you get lots of proteins found, and we call this the proteome or the matrosome when we talk about extracellular matrix proteins in particular. And these largely are driven by fibroblasts, which sort of stay quiescent within the stroma They are capable of repair pathways, so they only become activated in very controlled ways, such as wound healing, where they actively remodel the extracellular matrix, and then they're resolved via natural apoptotic pathways or switched off via epigenetic pathways. However, you can see here that there's lots of homeostatic signaling, but the physiological stiffness remains in range. You have very balanced mechanosignaling, and this is really here to provide structural support and maintain biochemical cues. Just to reiterate what's inside this matrisome, out of all of the proteome of the human body, about 1,000 proteins are associated with the extracellular matrix and ECM-associated proteins. About 270 of them are core structural proteins. So this is where the scaffold sort of analogy comes from, where they provide rigidity and support to tissues. And you can see that there's sort of several subgroups, and we can broadly classify the proteome into. So collagens, these are largely a fibrillar network. So they help with stability. We have proteoglycans. They do a lot of growth factor binding. They're also accounting for hydrating of certain types of tissues, and they account for differences in spacing and porosities. You have the big glycoproteins. Now, these are largely adhesive proteins. They're really, really essential in lots of sort of connective areas like basement membrane. You see a lot of things like fibronectins and laminins that are super important in the basement membranes of tissues. And then you also get other factors like intracellular proteins, ECM regulators. These are your proteases, so they break stuff down, help remodeling, like your MMPs. And you also get your cross-linking enzymes like your LOX, your TGM2s, TGM3s, as well as secreted factors. But all of these work together in a really nice biochemical way, and they really support the tissue. So they're super essential. Okay, so what happens in cancer? Well, basically, cancer completely hijacks this local system. And what it does is it shifts the balance from homeostasis in towards sort of fibrotic and excessive deposition of matrosomal proteins. You get a very sort of disorganized or altered organization in the tissue then because of that. And when you get this altered organization of proteins and architecture, you then get altered biomechanics. So you get this transition from a scaffold to a saboteur, which is one of the sort of central ideas of this talk, is that the tumor microenvironment, which is largely made up of this sort of extracellular component, is sort of driven by what we call the CAFs, the cancer-associated fibroblasts. They feed back along with signals from the head and neck sort of squamous cell core, and they're feeding into the local microenvironment, and you can see there's changes that occurs. You also get little pockets of changes. We call these ECM niches, and I'll talk about what we can see in head and neck ECM niches. But what I wanted you to get from this is that if the CAFs are the engineers, they're depositing ECM, what happens is once they've deposited this whole matrix is remodeled, it stiffens, and that has altered impact on both vascularity and changes in drug oxygen levels and everything. So what we've looked at what it does from a sort of schematic perspective, but how does it actually influence core behaviors? So this is quite a busy slide, but there's so much to talk about. Basically, extracellular matrix and changes in ECM will have impact on all hallmarks of cancer in particular. So it can largely drive invasion. So because it provides those aligned tracks, it acts as railways, and it basically means that cancer cells can migrate really rapidly across these aligned tracks. You also get quite permissive boundaries because of this, and those permissive boundaries promote survival by amplifying sort of local mechanotransduction signals. So because of those changes in stiffness, you're activating pathways, and these lead to pathways that increase proliferation and evade sort of cell death protocols. ECM can also act as a barrier, not just a barrier to cells, but a barrier to drug delivery as well. So you see that it has quite a big impact on immune modulation and the immune microenvironment. It acts as a physical barrier, preventing CD8 positive T cells into it, but it can also feed back signals to tumor-associated macrophages, and they have quite a reciprocal relationship within the head and neck tumor microenvironment, where they activate macrophages, which release MMPs, which activate CAFs further. So it's quite a distinct negative feedback loop between them. As well as that, I talked about drug delivery and the impact it can have. You can basically see that stiffened matrix alongside vessels. So this is where you have differences in the perivascular niche. You have a deposited amount of ECM here. This prevents drugs from diffusing in and out of the vessels. You can also have a completely juxtaposition where you have very leaky vessels. So you have lots of subsite differences in the vasculature at different time points as well. You can also look at differences in niches as well, and it has an impact on niches. And I've talked a little bit about perivascular niches, but there's also specific niches around tumors which can produce different proteins and have a different impact on perineural invasion and things like that. But this is a really sort of central to the story is the CAFs as the engineer activating and depositing these various ECM states, which we need to consider in our research. Okay, so I just want to point out that that's kind of what the extracellular matrix is doing within a tumor, but I just want to remind everybody that there are subsite differences that change tumor behavior. So when we consider things like oral, oropharynx, larynx, these will all have very different baseline stroma and very different vasculature patterns. And that's at the tissue level. When we go one step further and look at a micro level, then really the extracellular matrix is deciding what happens to the drugs when they penetrate. Whether we have alignment, porosity changes, that's all going to impact how we deliver drugs within that area. And this has impacts with clinical consequences. We're getting variability in response, whether that's to the actual treatment itself. We get toxicity versus benefit effects that we need to take into consideration. And we also need to consider the recurrence risk. So if you think about it, we're giving these patients all the same doses, the same regimes, in different ways, but they could all be experiencing the same sort of drug penetration pathway, but you're having different clinical consequences because of it, and it's likely to do with this micro-level biology. Okay, so in my research and during my PhD, I really looking at the extracellular matrix in sort of three axes. What do we know about it as far as its composition in cancer? What do we know about its architecture? And what do we know about its biomechanics in head and neck cancer? And there's a variety of ways that we can look at this and quantitatively study this as well. So first of all, from a composition perspective, there could be differences in the amounts of different types of proteins found within those tissues. So if we get more collagen, then we're creating more of a barrier, for example, and that can have an effect. For architecture, we tend to look at sort of three main features: alignment, porosity, and tortuosity. Alignment, we tend to view as looking at alignment of collagen, because collagen is the most abundant protein found, and that's the one that tends to get cross-linked and stiff. But we can also look at porosity between, so this is looking at the differences between the fibrils of collagen. And we can look at stress responses and tortuosity using elastic recoil and elastins. Another thing we need to think of is mechanics. So you can easily measure mechanics within tissues, and it's something that we've done. So we normally look at mechanics by way of stiffness, and that's a direct measurement of the collagen when we look at stiffness. And we can also look at tension and viscoelasticity, and we do that via rheology and rheological measurements as well. But stiffness we largely look at using atomic force microscopy, which is a physics technique, so you just sort of apply it to a biological setting. And there's lots of new microscopes that are more like bio-AFM adaptable as well. Okay, so what I have noticed and what lots of people are noting within solid cancer is that there are various types of ECM state and ECM phenotype that keep coming up, but you have different amounts of, or not at all. Now, all of these ECM phenotypes can coexist in the same tumor, can coexist in the same tissues, and can vary during subsite. So it's a very, very dynamic process and something that needs to be considered when we're looking at tissues. And the more we look at tissues in a spatial sort of capacity, the better we'll be at looking at these ECM states coexisting. So for the most part, in head and neck and solid cancers, you'll see three really core ECM phenotypes. The first is desmoplastic ECM. This is largely collagen-rich, CAF-driven. It often acts as a barrier to diffusion in drugs, but it really tracks for cells to migrate across, and cancer cells in particular to migrate across. We also have the sort of stiffened and cross-linked ECM. Now, this is where mechanics dominate over proteomics, and you tend to see this alignment and this cross-linked amplified mechanosignaling. So even if you don't see a difference in total collagen, you could see a difference in biomechanics just because of cross-linking phenotypes and changes in the cross-linking enzymes that are at play. You also see differences in biomechanical and transcriptional processes, again, which help evade apoptosis and encourage cell growth programs as well. Most commonly, what you see when the ECM is remodeled under stress conditions is senescent-associated ECM or therapy-associated ECM. Now, this is where the ECM, like I said, is remodeled under stress conditions. We're providing lots of toxic therapies. And what happens is you may get this persistent extracellular matrix phenotype that sort of is a memory and is retained within the tissue post-therapy that could cause a problem for ongoing reoccurrence. And like I said, key point here is that phenotypes can coexist and that this spatial distribution really does matter, and we need to consider it. Okay, this slide, I just want to highlight what you sort of see clinically. Most of the pathologists will probably see something like this. These are TMA oral cavity tumor cores, highlighting a stromal high, stromal low sort of pattern. So we can see this is the pattern of alpha smooth muscle actin, which is a marker of activated fibroblasts, and largely is found in desmoplastic stromas. You tend to see this sort of deposition of alpha smooth muscle actin. However, when you compare it to a low stromally active tumor, where you don't really see that alpha SMA staining. You can also see the linearity and alignment of lots of collagen just by looking at basic H&E. So here you can see all of these collagen tracks are lined really, really nicely alongside, and that's likely to help invasive processes. So it's easy to pick up. And what I want to point out here is that it's really great if the pathologists start talking about the differences in stroma and start recording it on their samples, so we can really see whether these patients have high stromal activity, low stromal activity, just by very simple staining. So that's largely how we look at composition. Very quickly, we use immunohistochemistry, but how do we look at collagen and architectural changes in samples? Well, you can use something called second harmonic generation microscopy. It's a label-free technique that uses the natural organization of collagen. When you laser align, it basically produces a signal. That signal is then converted, and you can basically see how collagen looks like through a tumor. So this is just a nice video basically showing you only collagen fibers because nothing else illuminates with that laser. But the good thing about this type of technique is you can do it on FFP, you can do it in your live samples. So you can get a really good readout that you can then quantify. So you get quantifiable metrics looking at straightness, and then you can link that back to other interesting phenomena like biomechanics going forward. So I just want to point out that you can look at these phenotypes as well. Okay, so one of the other core sort of ECM phenotypes that we see both clinically is a therapy-induced type of senescence or cellular senescence. And for those that don't know anything about senescence or the concept, it's an irreversible, non-proliferative, viable cell cycle arrest state. And this largely can emerge in the stromal components of head and neck cancers and other solid cancers. And the reason it matters from an extracellular matrix perspective is that senescent-like states can produce something called a SASP. This is a senescence-associated secretory profile, and basically that phenotype, and that basically produces lots of SASP factors, as you can see in this sort of diagram here. That feeds into the extracellular matrix and causes a lot of extracellular matrix remodeling and changing, including degradation, where it can be then taken up and eaten by macrophages and other things. So this sort of little niche sits in pockets of tissue post-therapy, and it's largely triggered by the therapies itself. However, it can be triggered from other insults like age, your microbiome, and immune signaling as well. But it's just something to point out that even though we've got these therapies, post-therapy, we still need to be thinking about extracellular matrix because it's still a factor when it comes to the senescent landscape in cancers as well. Some other extracellular matrix states that we're actively looking at, this is sort of some really preliminary data by one of the MSc students in the Hunter Group lab, whereby they were looking at the impact of cancer fibroblast-derived conditioned media and seeing if it would push macrophages into a more inflammatory or a more pro-tumorigenic phenotype. And just a very simple experiment of taking that conditioned media from the ECM that they deposit, adding it onto monocytes, you sort of see this M2 polarization, and we can see that we have an increased IL-10 expression, which correlates to a more M2 phenotype. And we're starting to see that in our other qPCR results as well, very preliminary. But this just goes to show that you can actually correlate changes in immune behavior just looking at either conditioned media from the cells. And we'll also be doing some substrate analysis, so the impact that monocytes have when they physically interact with different types of ECM. Just another point out that the head and neck squamous cell carcinomas also have other specialized extracellular matrix niches that also need to be considered that I'm not going to really talk about too much today. But you can see that we've got here immune ECM niches. These are largely shaped by, they largely shape as a two-way sort of interface. So immune niches are shaped by ECM cues, and then they simultaneously reshape ECM through other proteases, so, and cytokines and growth factors. So it's a bit of, again, like a feedback loop driving each other. We can also see there's other niche, specialized niches. So we have a basement membrane niche. So what happens is as the cancer cells are breaking through the basement membrane into the below connective tissue, into that stromal compartment, you see a really sort of rich protease interface, lots of MMP activity, lots of breaking down, and that's to help these cells migrate through. You know, we also start to see very sort of loose collagen and lots of fibroblast activity around the edge as well. You can also see what we call an inflammatory niche. That largely looks at sort of ECM that's maybe more wound-like, a bit dynamic. You tend to see a lot of cytokine-rich matrix, and it harbors a lot of cytokines as growth factors within that area as well. Other important niches to consider where the ECM is changing and is actively being remodeled are both the perivascular and perineural niches. Perivascular describes more of a vessel, blood vessel ECM interface, and you get a lot of compression factors and vessel compression surrounding here. So that's quite an interesting area that we need to really look at extracellular matrix properties in, because that'll have a big impact on how our drugs perfuse and how we deliver them as well. Again, the perineural niche, that has very specialized nerve ECM cues, but there's lots of perineural, as I say, there's not. So there is perineural invasion found in quite aggressive head and neck carcinomas. So it's interesting to see extracellular matrix changes around that perineural niche as well. And one more very interesting areas where the ECM is very different to other parts is at the invasive front of tumors. So not every bit of the tumor is migrating or invading, but in order to do so, what you need to have is some very specific cues and some very aligned fibers. So we do tend to see a very, very active cross-linking program around the invasive front. So I would likely see a lot of LOX expression in particular, but I would also see really, really aligned collagen at this front. Okay, so do we see the same thing that other researchers do within the Lhnc cohorts? And this is just some matrosomal changes that were observed in patient data here at the head and neck center. So this is data, unpublished data by one of the postdocs, Yetsi Montero-Bagara. And what she was found is that looking at an oral cavity cohort, there's lots of upregulated extracellular matrix genes. So you can see lots of collagen expression here. And it's really, really enriched in those sort of stromal matrix programs. So my take-home message from this is that ECM doesn't explain everything, but we're seeing extracellular matrix repeatedly visible at both the cohort scale. So that's gives me justification of why we should look at these sort of tractable ECM states and test mechanisms. She's gone a little further than this to look at a broader sort of niche landscape. And what she's found is that she can see ECM programs that are also comparable to an immune context. This is quite consistent, I'd say, with the idea that architecture, mechanics, they can all impact how gate traffic and residency of immune cells can all work. And stromal signaling can also shape immune tone within the same sort of microenvironment. And this is really highlighting that patient level signaling does exist, and we need to be aware of it. And our job in modeling those states is to see if we can convert them into measurable variables. And one of the ways to do that is to look at specific signatures within these cohorts to identify patients that are cold, excluded, inflamed with CAF high and inflamed with CAF low, because all of these will have a considerable outcome in what we should give them as therapies going forward. So my labs will be focusing on probably, or our lab is currently, and will be focusing on these three sort of broad niches. The desmoplastic stiff cross-linked ECM, which we said was largely a CAF-derived myofibroblastic ECM. We're also going to look quite a lot about the therapy-induced senescent ECM landscape. And again, looking at the immune ECM niches, but our group is largely focused on macrophage biology rather than any other type of immune biology. But the idea is that looking at all of these three will be done using quite a unified strategy. So we'll look at those three axes of composition, architecture, mechanics. We'll have quantitative measurements to read those out. We'll be able to model them quite efficiently in the lab. And then the idea is that over the coming years, we'll be able to test causality within those pipelines quite efficiently. So what have we been doing is modeling this extracellular matrix. And I've been doing this probably for the last 10 years in a variety of ways. So this research might be a bit higgledy-piggledy from all over the shop, but it's all led to a very concise idea that the extracellular matrix is super important for me. Okay, just to start with some of the gaps that we've been experiencing. Many of the models that we use to look at extracellular matrix within the lab, we use as a background material rather than a variable. So lots of those readouts will focus on bulk collagen abundance without sort of quantifying architecture, mechanics, stress, anything like that. Another gap we have are the platforms that we use. So we have great animal models to answer really great questions, but when we want to look at extracellular matrix, an animal model is no good to us. Completely different tissue structures. So it's not really applicable for this type of study. Obviously, ethical concerns, and there's lots of animal variability that might limit why we use them. The other issue that researchers have for extracellular matrix analysis is the insufficiency of ex vivo human tissue. It's critically scarce, and the reasons are really good for that. Number one is that your anatomy is related to your function. We can't go taking massive bits of your face out, even if we wanted to. You need it. You need it for a lot of things: eating, swallowing, speaking. It's your whole personality. So it's very difficult for us to get tissue. Especially on the large scale, and especially to do things like drug development experiments or drug permeation experiments. We can't get the ex vivo tissue large enough in order to do this. As well as that, lots of the in vitro models are quite simple. What they don't do is have the really mature ECM structures that is required. So the fibrillar ECM and the 3D ECM that we see, you don't really pick that up a lot with some of the 2D cultures that you can get. So modeling it in the lab is very important. And I think I've got the toolbox to do it quite efficiently. I have a lot of experience of developing subsite-specific mucosa. So I've developed models from different sites of the mouth, developed and isolated fibroblasts from different sites of the mouth. I've become quite proficient in human fibroblast-derived ECM techniques. So I can deposit very thick 3D native matrices that the cells basically do the hard work. I just have to keep them alive in a lab. I could also look at building 3D tumor models. So I have a lot of experience with transwell models, hydrogel-based modeling, steroids, and I'm branching now into the more organoid, humanoid-based modeling, which I think is more important for this type of work. And I also have got some great platforms, or I've used them as platforms for therapeutic testing with some really, really good quantitative readouts, which I think are quite valuable if we're going to look at this translationally as well. So what I'm quite well known for is these native extracellular matrix platforms. And how this sort of works is we take the fibroblasts from the patient tissue of origin. Ideally, this would be matched normal and cancer fibroblasts from the same patient. But I have a culture technique where they deposit matrix over a period of time, and you get a nice mature extracellular matrix. And these are just H&E images of that matrix. So we've got the normal matrix here and a cancer matrix, and you can quantify how much protein comes out of it, and you can also have a look what's in it. But you can already see just by looking at it that it varies in composition and thickness, just from normal to cancer. But there's lots of other features that these can be used for, and they have been used for. So what we can measure using them and using that sort of axis that I was talking about, we can look at the composition, and we can do that a lot with proteomics-based analysis. We can also look at the architecture, and for that, I like to use the second harmonic generation, where you see the alignment of fibers from normal to cancer, where you get more invasive tracts, more aligned fibers, thicker fibers as well. We can already see that the matrix is changes in its biomechanical properties. So the cancer-associated fibroblast matrix is stiffer than the NOF matrix. So we get a difference in stiffness profiles that's easy to quantify. You can also see some of the biochemical changes from normal to cancer as well. So this is just a secretum or cytokine analysis looking at, I think it was 105 different human cytokines or something like that. And you can see that the CAF secretum, there's lots more black dots, which is telling us that the secretums are far more active and depositing a lot more in these matrices. Okay, another thing that you can use them for, and lots of people use cell-derived matrices at 7-day culture, but they don't support epithelial development or full thickness models, whereas these longer term ECMs do. And I can make normal models out of them. I can make CAF-derived matrix with normal cells, and you can already see that these are the same cells here. The only difference is the matrix, and I'll show you on the next slide why that's important. You can also put cancer cells on top and build sort of full thickness cancer models. But what's very interesting about these, even though they're a 2-cell model type, they can be kept in the lab for up to 6 months. So this is just, these are only NF2, but I can keep these alive for months in the lab. So you can look at long-term changes, very dynamic changes in the ECM over time, which you won't be able to get in short-term cultures. Why does time matter? Well, it really does matter for therapy-induced fibrosis and that therapy-induced senescence profiling that's happening. So this might be occurring weeks, months after the disease is there, these little recurrent niches that are only senescent pockets. And we could potentially be able to pick up those just by tissue engineering. The other thing we're able to do is look at differences in behaviors of different cell types using these matrices as scaffolds. So this is an example of me using it to look at cancer cell behavior. So this is a tongue carcinoma line, and I've put it onto a CAF matrix, and I've basically measured how the cells move across the tissue. But you can look at adhesion rates, migration rates. This is an example of population doubling here. So if I put normal cells on a normal matrix and I put the same cells in a CAF matrix, we see an increase in population doubling, a significant increase as well. And if we add a tongue carcinoma line on a normal matrix or to a CAF matrix, you can already see that the CAF matrix, these cells are dividing much more rapidly. So they're really great substrates to look at a lot of interactions. Another thing that I want to point out is that this is where it really drills home that sort of causal logic of in its simplest form of why I want to look at the extracellular matrix. So here we have the same epithelial cell line, and this is considered a normal buccal cell line. The only difference is the matrix that I've put it on. But you can see absolutely the changes in the epithelium, a lot of atypia, a lot of loose connections. I've stained this up, and it's a completely different epithelium to this, and these are considered normal cells. So if changing only the extracellular matrix and that's driving invasion, morphological differences, survival differences, drug differences, then the matrix is becoming quite an upstream lever and potentially a very plausible biomarker for us to consider. And it sets the stage for developing matrices that quantify this in very clinical contexts as well. Another thing one of my students has been looking at, and again, some preliminary data, is how can we sort of look at stiffness and really analyze the cross-linking machinery in head and neck cancer. So I'm quite interested in Lhnc enzymes, and how that sort of fits in this whole paradigm is that Lhnc mediated cross-linking, it provides covalent cross-linking to collagen fibers, bringing them closer together, making them stiffer. And then you get this stiffness that activates these pathways in tension, biomechanics, And all of these promote adhesions, cell motility, and basically the ability for cancer cells to migrate really efficiently. So I'm quite interested in how LOX is changing in the extracellular matrix of cancers. So we've looked at this at a qPCR level and then into Western blot, and you can see that in particular LOX was found significantly higher in the CAF compared to the NOF. And when we looked at this at the protein level, although this hasn't been quantified because I had to do, I must admit, a little bit of post-image editing here. The student had quite a bit of background on these Western blots. So it's not publication ready, but I just wanted to highlight that these are patient-matched NOF-CAFs, and you can see the difference in LOX expression quite clearly, with the CAFs more abundantly expressing LOX enzyme than the NOFs. I will caveat that with this doesn't tell us anything about activity, just presence of the LOX enzymes, but some really nice prelim data to say that LOX could be an interesting target. And I think this was an OPSCC cohort, so this is oropharyngeal cohort, which was quite interesting. Another thing that's happening within my group is that I currently have some proteomics underway. So I'm just going to present the rationale rather than any actual results. So I'm going to be taking tumor tissue from different subsites of the head and neck, as well as tissue engineering different ECM from different subsites of the head and neck. And I'll be using a mass spec quantification technique. So this has been pioneered by Professor Sheltner at our proteomics facility, and we'll be using a quantitative detergent solubility profiling method, which basically is going to take the most insoluble fractions and then solubilize it and solubilize it and keep solubilizing it until we go from the largest fractions to the very, very smallest. And I really want to look at the collagen and LOX profiles of these to see what we can find, not only in matched patient differences. So do we see differences in the subsite of matched tissue? And does that vary then within anatomical subsite of the head and neck? So there's quite a lot of interesting things going on there. And I'll be looking forward to getting some of that proteomics back. This is where I'm just about to plug my Xenostroma platform. So I've just been given a bit of money to push this forward. And the idea is that I've developed these ECM platforms, but I need to make them more high throughput. And I'm largely looking to do this for oncology and aging research purposes. What Xenostroma is I take my native extracellular matrix material and I'm producing hydrogels that are suitable for high throughput testing modalities. But alongside making these biomaterial hydrogels, I will also be developing a matrix sensitivity index. So Xenostroma is developed from both matched normal and CAF-derived matrix, and these are basically patient hydrogels that I can make. But I'll be using them in either an organoid or spheroid pipeline to really look at the sensitivity of drugs or of our matrix to different types of drugs. And this is basically a way of making my matrix context quite quantifiable, and no one's really got any idea of how to look at measuring the response of ECM-related or stromal-related genes within this context. So I'm hoping that I'll have quite a lot of information. I'm stripping on myself a bit because I don't know how much I'm allowed to say or not say. So I've been told to keep things very, very sort of surface level. But I'll basically be taking a drug panel of both clinically relevant drugs and maybe some novel drugs and adding them to matrices at different subtypes. So I'll have a normal baseline, I'll have a cancer subtype, but I'll also have a senescent or aging subtype. Run my drugs through them, I'll have multiple readouts, and the idea is that I'll be able to create an analytical pipeline that will quantify how those drugs behave on the matrix. And we'll be able to potentially look at whether patients will respond or have therapeutic resistance just based off this stromal profiling. But again, this is very much the infancy, and this is what I'll be working on this year. So if anybody has any drugs that they're, especially any stromal targeting drugs, they're very interested in finding out their activity, let me know. Okay, so this is just very briefly to summarize. I want to just describe how I've told you about how it goes from really a scaffold to this saboteur, this active driver of extracellular matrix remodeling and driver of cancer behaviors as well. I want to reiterate why we use this sort of site-aware ECM states and why that's so important, and how we can model that in the lab. And the idea is that this should be translatable for us to provide biomarker stratification, therapeutic stratification, and whether we need to start using combinational therapies or adjuvant therapies that maybe target the stroma in certain cases, rather than just the standard treatments we have currently. So take home, ECM is not just a passive scaffold. It shapes invasion, immunity, and therapy response. We need to think about how we use controlled, clinically anchored ECM models that are representative of our local cohorts here at Liverpool and of the disease in general. And we need to come up with architectural reasons, stiffness reasons, compositional reasons, cell state reasons of why the ECM is such a hindrance to us. And the idea is that if we can look at these ECM features, then they can be exploited, and whether that's as biomarkers or whether that's as therapeutic address, and that's something we need to consider. So as always, if you have any pre and post therapy annotated samples that really look at matched stroma or biomechanics, any clinical metadata that's got anything to do with clinical biomechanical data, please let me know, because the literature is not very good. And I'm also interested in testing lots of different stromal therapies. So if you have one that you're developing or you're interested in something in particular, please let me know, because it can be added to the drug list. And then if you have any of the following interests, please get in touch. Apart from that, I'd like to answer any of your questions.
Thank you very much, Amy. And there are quite a few questions. I jotted this down a little bit as well, but there was a question from Daniela Elena Costea about the differences in the ECM, I guess, between HPV positive and negative. Extrapolating what you've said in terms of the behavior of tumours and response to treatments, you would expect quite a big difference between HPV positive, which respond very well. So could you characterize the tumours just looking at their microenvironment?
I'd like to be able to do that for the most part. I think the reason why you're seeing differences in the HPV positive and negative is largely to do with the stromal differences as well. I don't think you see the desmoplasia in oropharynx tumours like you do in the oral cavity. You tend to see the tongue desmoplasias as being more active there than you do in the oropharynx. Now, is that because you have less stroma, are we seeing better outcomes? And that's really what I need to find out, and that's what I'm looking into specifically. What was the other question, Richard?
Oh, there's a list. So I think that's a reasonable question. Yeah. So the next question was from Peter Waitman, and Peter said our group has infrared imaging system that can detect molecules like DNA, fibronectin, collagen. I'd always wanted to use it to measure collagen alignment, but not yet had an application. Would that be useful and interesting?
Yeah, absolutely. I think any way to get better readouts. Second harmonic is great, but you're using a laser alignment to look at it. So you can sometimes destroy the tissue whilst you're doing it, especially especially my tissue engineered ones that are very, very thin. In thicker tissue, that's not a problem, but you could disrupt the tissue using second harmonic, and we largely look at that from sections. So looking at something live using the infrared, yes, very interesting. I had also had somebody else say that they could look at collagen through X-ray. an X-ray diffraction methodology. So there are lots of ways that we could do it in patients that's clinically relevant as well. So yeah, if you want a model to look at, I'll happily give you one.
There's a question from Lucy Bosworth. Really interesting, thank you. Can you tell me what the cells are being grown on? Is it a tissue, culture, plastic, or a biomaterial scaffold? Do you find that the substrate affects cell response?
So my cells aren't grown. So in 2D, they're grown directly on plastic. This is a really big problem in vitro scientists have, I think, Lucy. So we isolate the cells, we shove them onto plastic. It's a really artificial, stiff environment. We might be altering the epigenetic profile of those cells almost immediately as soon as we put them on plastic, which isn't a good idea. So we've had this discussion internally. When we next isolate cells, the first thing we are going to do is isolate them onto a collagen 4 membrane, we think. For these cell-derived ECMs, the cells are put onto a pore-lined transwell insert. So there's no gel, there's no gelatin, there's no nothing. The cells are seeded on there and they basically deposit their own ECM over time. We don't need to add any matrix layer. When you do any sort of analysis, I can remove the whole of that matrix layer from the transwell so that it's sitting on whatever I want it to sit on. If that answers your question. So the idea is no, the cell-derived matrices are just on a insert. There's nothing coating them at all. However, I do think there is relevance to coating on 2D cultures, especially when you're first isolating fibroblasts.
Great, and I think we'll make it the last question because we're just approaching 8 o'clock. There's a question from Josh Hattersley. Great talk. Do you know how similar the proteomes of your matrices made from your CAFs are that you go on to use?
So I've done it, I haven't done it from a bulk proteome, that's what I'm doing with the mass spec analysis, but I have looked at very specific protein expression, protein abundance using Western blot. And you do see differences in the abundance, definitely from the NOF and the CAF-derived ECM. So I looked at alpha smooth muscle, I've looked at various types of collagens, collagen 1, collagen 4, and these are all done via immunohistochemical staining. And you definitely see that. You also see it from total protein from the lysates as well. So yeah, you definitely see more being deposited and it's much more abundant.
Amy, that's absolutely great. Thank you very much. We're approaching 8 o'clock. I'm going to let people get on with their evenings and catch up on traitors or whatever it is. You could have done a little traitor analogy for the extracellular matrix, couldn't you?
Oh, do you know what? I'm not watching traitors, but now I feel like I should.
Well, I get forced to watch it by my kids, believe it or not. What I'm going to do just to finish, as well as, of course, thanking Amy, and you've had a great audience. There's been standing room only. There's been 40 people in the lecture, so that's great. I'm just going to, just to finish off, just share the flyer for next week's talk. Hopefully we can see that. And next week's talk is Dr. Gemma Cherry, who's an academic clinical psychologist who's working in this area of quality of life in older adults with head and neck cancer. So thanks, Amy. We look forward to Gemma next week. Thanks, everybody, for logging in. And please do spread the word. We're not going to outstay our welcome. This is going to be done within an hour on 5 or 6 consecutive Tuesday nights. Then we'll all be done and it'll be a whole year before we come back. So fill your boots while we're in the bleak midwinter. And Amy, you've brightened up our evening. So thank you very much. And thanks for a great talk. Thank you.
Thanks all.
Bye.